Hey everyone, welcome to our video about DNA mutations. These are types of mutations that occur due to a change in the base pair that is made during DNA replication. Um, DNA replication occurs when new cells are made, and when you make new cells, that could be for just making regular cells in your body, that's called mitosis, or making reproductive cells, and that's called meiosis. And if some of these mutations occur when you're about to do meiosis or make an egg and sperm cell that's going to be used for reproduction, this could cause a child to be born with a genetic condition. And so these mutations can cause genetic conditions. But these are all mutations that relate to a specific change in the DNA sequence of a person who might have one of these conditions. So, the first type we're going to talk about is called a missense mutation. And this is a type of mutation that causes a change in just one DNA base pair. It's basically substituting one base for another, which results in one amino acid being substituted for another when somebody goes to make a protein, because DNA is actually the instructions for making proteins. So if you change DNA, you're going to change what proteins you get. So in this one, you can see CAT would be the typical sequence here, but in this one, there's been a mutation. Instead of an A, you now have a C. And so that actually would go on to change the protein that gets made. And that can end up causing problems for somebody. So a missense mutation just changes one DNA base pair. And an example of that is sickle cell disease. So somebody who has sickle cell disease has red blood cells that are formed a bit differently. They are flattened into what we call a sickle shape. So instead of being kind of these plump round blood cells, they are these kind of flattened sickle shape blood cells, and they do not carry oxygen as well. People with sickle cell disease might need blood transfusions, or they might need oxygen. And it all just occurs because of one base pair getting changed, one getting substituted in a missense mutation. So that A got changed to a T. Basically, the DNA didn't get copied correctly, and that's a missense mutation. Next, we're going to talk about a nonsense mutation. So, a nonsense mutation is when a base, like a G or a C or an A or a T, a base, nucleotide base gets changed, but that change in the base causes the protein to stop being made completely. So you get this like really short protein instead of where you would have had a nice long protein, you get this short protein. So that's again just because of one individual base being changed can cause this nonsense mutation that causes the protein to be stopped and causes a person to have a shorter protein. An example of that would be cystic fibrosis. So somebody who has cystic fibrosis has trouble breathing, can have digestive problems. They might need to be on medication, have breathing treatments. You can see there a nebulizer, that's a breathing treatment. It's kind of like an inhaler, but it gives you more medicine than what an inhaler would give, and it takes a little bit longer period of time. This vest that this patient is wearing actually is called a percussion vest, and it kind of provides the effect of somebody hitting your back while you're coughing to help you loosen up your mucus. This often causes people to die when they're at a young age. And so it basically just causes you to build up fluids in your lungs and have just less ability to be able to breathe properly. Again, it can also add mucus in your digestive system that can cause digestive problems as well. But all of that occurs just because a nonsense mutation caused a DNA nucleotide to change and made a protein become shorter. The next types, there's actually two types of frameshift mutations that we're going to talk about. This is going to mean that you're adding or losing DNA bases, and when you do, the entire sequence of DNA gets shifted by one letter. And when you shift it by one letter, you actually end up with entirely different amino acids being made to go into a protein. So if you completely change your amino acids, you definitely completely change the proteins that are made by this code of DNA. And so you're going to change what proteins a person has, and that can cause some serious problems when we're talking about genetic differences and people having genetic conditions. So, one type of frame shift is called an insertion, and this basically adds a piece of DNA. It just basically adds in one extra nucleotide. So, just adding in one extra nucleotide here. So, this is not a real protein, but let's just imagine it said CAT, 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 CAT. Well, if you add an extra A in there, now you've shifted everything over by one, and you change the entire sequence of proteins that are going to be made. So just adding in one extra DNA nucleotide to this piece of DNA changed everything about that DNA and changed which amino acids it would call for when making a protein. An example of that is Lou Gehrig's disease, or ALS. A famous person who has this disease is Stephen Hawking. He experienced some dementia, muscle weakness. Medication is needed for these patients, physical therapy, a lot of times comfort care. And this can end up causing somebody with ALS to have a much shorter life expectancy than a person normally would have. Actually, Stephen Hawking lived a fairly long life for somebody who had Lou Gehrig's disease. Most people's lives are much shorter if they're diagnosed with ALS or Lou Gehrig's disease, but it's all caused by just one extra nucleotide being added in, and that changed the entire protein that was made in Stephen Hawking's cell. Basically, what happened there is that that protein was needed to make myelin, and that myelin stuff helps these nerve cells send signals to muscles, and without strong myelin, the muscle does not receive the signals and your muscles start to waste away. And that's why you see patients with Lou Gehrig's disease end up in a wheelchair. Their muscles have not been able to be stimulated, and they're not able to do walking, talking, breathing on their own. Another type of frame shift mutation is deletion, and this occurs when you don't add a piece of DNA, but you actually remove it. So by removing just one nucleotide here, you've changed the entire sequence of nucleotides. You've changed the protein that is going to be made from that DNA just by removing one single nucleotide, removing one letter. And an example of that is Tay-Sachs disease. Tay-Sachs disease is again caused by just removing one nucleotide, which changes the entire protein that will be made by that DNA. And it results in children, typically children, having lipids in their brain or fats in their brain cells, way too much fats in their brain cells, and those lipids go to the lysosomes. Those lysosomes can end up bursting or rupturing, and it causes, because it's in the brain, a lot of weakness, lack of coordination. Patients might end up being blind because of Tay-Sachs disease, and typically these patients do not live a full lifetime. They have an early death due to this buildup of lipids in the brain. So what I want you to take away from this little lesson about mutations is it really can just take one change, like deleting a nucleotide, adding a nucleotide, having just one change, swap one for the other. It can just take one nucleotide difference to cause an entire group of proteins to be formed differently, and then people end up having physical effects because of that. So these are DNA mutations. Later on, we'll talk about chromosome mutations, but these ones are DNA mutations. Thanks so much for listening.